Okay, well, I think it's 12:05, so we should go ahead and start. So welcome everyone to the Melbourne Integrative-Genomics seminar. Lachlan was always scheduled to be our speaker at this time, and originally intended to be in person, but obviously we've switched to online, which has the advantage that a larger number of people, as you might have heard me say, there's more than 200 people registered for this seminar and 128 attending at the moment, so that's great. That probably means many people outside Parkville and maybe even outside Australia are joining in, so welcome. We did have a program of seminars every other Friday throughout this semester. Obviously everything's in a little bit of chaos at the moment, and where for the MIG people on this call, I want to have a little Zoom catch up later to discuss the future plans for the seminar. We have a little bit of disagreement about you know, to what extent we want to defer speakers rather than having further online seminars, but I think it's a good sign already that so many have joined us for today. Lachlan is an old colleague of mine. He did his undergraduate in maths at ANU and then a PhD in bioinformatics at the Sanger Institute in the UK with Richard Durbin. And then we were lucky enough to recruit him. I was then at Imperial College, and I still remember the outstanding interview he gave, and we recruited him to a research fellow position at Imperial College that before we moved to the University of Queensland. And now again, we've been lucky to recruit him to the University of Melbourne, where he's recently been appointed a professor in the Department of Microbiology and Immunology in the Doherty Institute at Melbourne. So that's great that he's been able to come to Melbourne and that he's been able to present this seminar to us today on a very topical subject. So please go ahead, Lachlan. There will be a Q&A at the end, and Lachlan will manage that. There is a Q&A box, and you can type in questions there, which is what we prefer, but there is also a raise hand option. And if there are no typed in questions, Lachlan will answer the raised hands at the end, and you can just, then you get unmuted and you get to ask your question. So thanks very much, Lachlan, and please go ahead.
Thanks very much, David, for that kind introduction. I'm going to turn on my webcam just in case I gesture. It sometimes helps to give the talk. Yes, this is the first time for me giving a talk via a webinar, so be interested to see how it goes. And this project's obviously a collaboration with a number of people at the Peter Doherty Institute and at Vidril, and I'll acknowledge everyone at the end, but it's a collaborative project. So my abstract actually aged really quickly, unfortunately, and this slide's aged since yesterday as well. So this is just basically to say that, as we all know, COVID-19 is a huge problem. There's a large number of cases. It's now cleared 1 million apparently, and there's substantial mortality associated with that. Now, most of the SARS-CoV-2 sequencing that's being carried out is direct from patient sample. And it's very hard to sequence native RNA of the virus direct from patient sample. And so most strategies are employing a tiled amplicon approach. So this was a figure that James Ferguson posted on Twitter the other day, showing how he's managed to get a tiled amplicon approach working on a nanopore fungal, which is like a cheap disposable nanopore chip. And so the design here consists of you can see just here, there's 14 amplicons tiling across the almost 30,000 base genome. I have to be careful not to say base pairs because obviously it's a single-stranded mRNA genome. And this also shows the coverage that you get from that. So you can see that you get spikes in coverage where the primers overlap, as you might expect. So you get kind of an even background coverage and some spikes. And this is perfect for finding mutations and obviously for epidemiological tracing of cases. It's good for that, but it provides no information about the transcriptional activity of the virus. So SARS-CoV-2, as we all know, is a recent introduction into humans. It's kind of a recent virus. So this is work carried out by Leo Featherstone and Sebastian Duchenne at the Peter Doherty Institute, colleagues of mine. So they downloaded high quality sequence data from GISAID and used Bayesian approach with BEAST to basically calculate a kind of a high confidence tree. And from that, they can date the emergence or the most recent common ancestor of all the SARS-CoV-2 that's been sequenced to about the end of November. So it's very recent emergence. It's obviously for society a terrible thing, but it's also scientifically very interesting to see this virus mutate and expand so dramatically around the world, as there's no immunity to it in humans, obviously. So why do direct RNA nanopore sequencing of a virus? So there was this paper, conveniently, which came out in Genome Research towards the end of last year, but before SARS-CoV-2 emerged, where Viewegger et al. Did the first direct RNA nanopore sequencing of a viral sample, and they used HCOV-229E, which is another coronavirus infecting humans. There's two lines here, but I'll just focus on the blue line. So this is basically a coverage plot. So compare this to the coverage plot that we saw from the tiled amplicon strategy. This is the coverage plot you get from direct RNA sequencing of a coronavirus. And you can see that there's a number of features you see here. Basically, you see this stepping down on the right-hand side, stepping down of the coverage that you can see it jumps down and steps here, and that's indicative of what we call subgenome length mRNA, and I'll explain that in a little bit. And basically, you also see that so you have to remember direct RNA sequencing sequences from the 3' end because it's primed on the poly A. And so essentially, the further you move away from that, the coverage decreases, and also some of these longer ones have probably less expression as well. And so they showed in their paper that you could identify different subgenome length mRNA. So they identified 7 subgenome length mRNA species. And these arise from a process called discontinuous extension of minor strands, but can kind of essentially think of it as joining up the leader sequence, which is the 5' end. There's a sequence which is thought to initiate translation. So that leader sequence, in order to translate the proteins towards the 3' end, needs to be basically put in front of every one of these open reading frames on the 5' end. So that's what this discontinuous extension of minor strands process does, is it joins up this leader sequence with each of the open reading frames on the 3' end. And they also showed in their paper that they could detect 5MC RNA modifications using Tombow. So we followed a similar strategy and used material. As I'll explain later, George Tararara did the sequencing, but initially he sequenced RNA extracted from the viral particle, from the virion, and we got modest coverage from that sample. And then later on, once enough material had been grown up in cell culture, we obviously had access to much more RNA, and then we could do direct RNA sequencing from the cell culture derived RNA. And I'll compare and contrast those two in this talk. I also want to point out there's been 2 other papers that I'm aware of that have since come up on bioRxiv looking at direct RNA sequencing of SARS-CoV-2. And they're both excellent. I encourage you to go and look at them if you're interested in this area, and they focus on slightly different aspects of the data. And each of them, as we have, has made the data available too. So it's possible for anyone who wants to go and download this data and analyze it themselves. So this slide indicates the genomic structure of, of the genomic structure of SARS-CoV-2. So we see that, as I mentioned, there's a number of open reading frames across the genome. So these are indicated by the different colored blocks. So on the 5' end of the genome, we have the open reading frame 1A, 1B. What I should say that 1B is translated, there's a stop codon at the end of 1A, and 1B is then translated by a slippage through that stop codon. And you can see that 1A and 1B actually encode for quite a number of different proteins. So that's kind of, as I understand it, translated in a block, and then there's proteolytic cleavage that separates these different proteins into their different parts, and then they go off and do their separate functions. So there's around about, I think, 26 proteins in total encoded by the genome. And so all these NSP proteins in the 1A and 1B open reading frames, and then the structural proteins on the 3' end of the genome. So S is the spike protein, is the envelope protein, and then a number of accessory factors as well. And one thing you'll notice is that this is a little bit more complex than the annotation that we saw for the HCOV229E genome. And so this, so I took that figure from an excellent paper where they looked at protein-protein interaction map between human proteins and SARS-CoV-2 proteins. And so they did extensive characterization of the proteome of SARS-CoV-2. And so also from that paper, they basically looked to annotate the function of all these proteins. And so that's what you can see on the left-hand side, and look at the similarity with the previous SARS coronavirus. And so you can kind of see the, the annotation of the envelope here, the membrane glycoprotein, for instance. And one thing you'll notice is that, and I'll come back to this later, is that ORF10 is an interesting open reading frame in that it has no known function. And in fact, the protein which is supposed to encode doesn't match anything in the databases. So it's unclear whether that's actually expressed at all and whether it's important. So in this paper, they did some immunostaining, which is shown on the right-hand side here. And so you can actually see, so at the top, we've got the NSP proteins, and then down the bottom, we've got the other open reading frames. And so you can see in the right-hand side that they actually saw some evidence of expression of this ORF10, which is, unlike any other known protein. So at the protein level, there seems to be some evidence that it is actually being translated into protein. So, okay, so this is now moving on to our data. So obviously the first thing we did was try and replicate the coverage plots that the HCOV229E paper had. And so you can see that coverage plot. We split it into all of the reads, and then the reads which have this leader sequence. So the leader sequence is important in that we only expect that those two leader sequences will be translated into protein. And so you can see there's substantially less of those, but you can also see that this laddering effect is much more pronounced in those. And so these are thought to represent the subgenome length mRNA, which basically corresponds to kind of putting that 5' leader sequence in front of all of these 3' open reading frames. So yeah, I mean, this is interesting, but it kind of only shows you part of the story because it's kind of just showing you everything together. And what we want to do next is to be able to split out each of these subgenome mRNAs, look at their expression levels, and look at other characteristics of them. So we wanted to investigate different strategies for clustering reads into transcripts. On the right-hand side here, we can see the coverage for the virion data set. So the left-hand side is the RNA derived from growing up the virus in viro cell culture, which is a cell culture which is amenable to infection and which the virus can replicate quite quickly. And then the right-hand side, this was the initial data set that we collected, which had about 10% of the reads mapping to the viral genome as the cellular data did, so less coverage to start off with. But the other thing we observed was that the reads mapped with the leader were a small proportion of the total. So it seems, so most of the reads, in fact, we got fairly few with the leader sequence. Most of the reads were leaderless, which is what you would expect, I guess, if you're sequencing just the full length mRNA molecule. And so if that would kind of be what you would expect if there was little RNA-dependent polymerase activity actually expressing these subgenomic mRNA. So that's perhaps not unexpected that the virion is kind of in much more of a nascent state. So going back to this idea of trying to bin out the different subgenome linked mRNA into, so bin out the reads into the different clusters, we constructed this linked histogram of all of the reads which had a leader sequence. And so then we've annotated the link that we would expect to get from the different classes of subgenome linked mRNA. So ORF10 is the one on the left. You can see, so the top, I should say, is the cellular derived RNA after growing up in the virion cells, and the bottom is the virion RNA. And you can see here the coverage is much lower for the bottom plot, which is a compound of the fact that we've got less reads overall, and also a fewer proportion of them had leader sequence. And so you can see in the top plot that this N protein, or this N open reading frame, I should say, has the most reads mapping to it. And we see a few of the spike proteins. So the spike is one of the longer 3' open reading frames, and that seems to be expressed at lower levels, or is perhaps slightly confounded by the fact that just statistically you may not sequence the full linked mRNA if the molecule, say, breaks while it's going through the pore. So we're not quite sure that that's as low as it seems. And then on the left-hand side of this plot, we've got this mysterious open reading frame 10. And it looks from this plot, you would conclude that open reading frame 10 is not actually expressed. And the other preprints have concluded this as well. In particular, the preprint from Korea also concluded that ORF10 doesn't seem to be expressed. And then on the bottom of this, on the virion, you see, we also see of those that do have a leader, which isn't many, we see most of them at this N open reading frame, and there are a few of the others as well. So based on this linked histogram, we can then, the peaks are quite well separated. So we used what's called a smooth Z-score clustering approach to assign reads based on where they lie in this linked histogram to the different bins. And then when you separate that out, you get this coverage plot. So each line here is the coverage of a different bin, essentially. And you can see, the laddering is obviously very apparent, as you would expect. This is a log scale on the y-axis. And there does seem to be a little bit of activity here on the left-hand side. So we expect these leader-containing reads to only really contain the leader. There's a what's called a transcription regulating sequence around 65 base pairs. So essentially that sequence should be the one that gets joined up with a matching transcription regulating sequence on the 3' end, but we see there's some leaky reading through that down the bottom here. Okay, so that length-based binning approach is kind of okay, but binning based on a histogram is kind of obviously going to be error-prone, particularly if the peaks were closer together. So we've been exploring different clustering approaches, and the one which I'll present the rest of the results on is based on this strategy. So essentially, we're trying. So you can obviously cluster reads in a variety of ways. You could just do a pairwise mapping and look at identity between the two, for example. But we wanted to do something which is kind of quick and also reflective of the structures we're trying to capture. So we used a Cigar string clustering approach. So the way that works is that you map the reads to the advantage of this is it's a single pass. It's very quick as well, and so it's very easy to process lots of data sets. You map the reads to the reference genome using Minimap2, and then essentially from there on, you don't look at the read at all. You just look at the Cigar string if it's mapping to the reference. And so you retain reads which start within the first 100 base pairs. So because we only want those reads which have this leader sequence essentially, and which kind of end within 100 base pairs of the reference end. And then we inspect the Cigar string essentially to look for long runs of ends where there's reference but there's nothing to map it to in the mRNA. And so we look for the longest stretch, which is greater than 1,000 base pairs. to find the breakpoints, we record the 5' and 3' breakpoints, so where the N starts and stops in reference coordinates. Once those breakpoints are identified, we find the upstream, the first start point of the upstream open reading frame, upstream of the 5' break, and the first, and if there is no open reading frame and it's just a leader is the only thing upstream of that 5' break, then we annotate it as a leader only. And then we also find the first 3' downstream start site of open reading frame. And we have a 10 base pair tolerance as well to allow for some error in the nanopore reads and some alignment error as well. And then essentially we just bin reads based on their assignment of upstream and downstream open reading frames. So anything with the same upstream and downstream open reading frames will go into the same bin. At the same time and simultaneously, we basically have a sparse matrix which keeps account of how often each 5' and 3' junction is utilized, and we just update that sparse matrix with the counts of how many times we see each of those combinations. And so this is what we see as a result of this second part of that, which is the breakpoint clustering. So I'll just focus on the left-hand slide of this, left-hand part of this slide. So on the x-axis, and up here we have the different colors indicates the different open reading frames, and that corresponds, so think of the x-axis as corresponding to the 3' end of the molecule. And so you may not be able to read these numbers, but essentially these are 3' coordinates starting from just about 20,000 bases going up to the end of the molecule. And then similarly, we show the genes on the so the 5' breakpoint is on the y-axis, and so that's the start of the genome, so going up to 100. And then here we've indicated in yellow where this leader sequence is. We've also indicated with these white lines here where the transcription regulatory sequence is. So this is a short motif, which is thought to promote this joining between the two ends, essentially. So what we would expect to see is most of our breakpoints should occur at the 3' end on the vertical line, sorry, on the horizontal line, and at the 5' end on one of these vertical lines. And that's indeed what we do see. So I should say that to identify all of the positions of the TRS, we know where the location of the 5' one is, and so we built a position weight matrix using all of the reads mapping to that part of the genome, and then use that position weight matrix to scan the rest of the genome for these transcription regulatory sequences. And so that's where we see all these ones on the 5' end. And then in terms of the heat map, the actual value is the log of the number of times each breakpoint is used. And on the y-axis, it's a base pair or base resolution, and on the x-axis, because it's only 100 bases, we can do that. But on the x-axis, it's binned into 10 base pairs. And so what you see is kind of what you expect. Around 65 base pairs is where there's a high activity, but there is some kind of variation here as well. So although this is log scale, it's very much concentrated here. And then you can see on the y-axis where that 3' breakpoint is joining. So you can see this orange gene here is this N open reading frame, which you saw was the highest expressed. And so therefore, as you would expect, there's the most breakpoint activity just immediately upstream, exactly where the transcription regulatory sequence is, which then joins this leader sequence on the 3' end up to the 5' end of this N open reading frame. And so, yeah, you can see basically there's activity in front of each of these open reading frames on the whole, although some of them don't seem to have a recognizable transcription regulatory sequence. For instance, this ORF, this E ORF, we couldn't find a TRS immediately upstream of that. Okay, so that's the left-hand side. Oh, I should say with ORF10. So for ORF10, if we were looking for activity, what we'd see, if there was activity, we would expect to see a TRS in front of ORF10, and there's not. And so that, would suggest it's not really expressed, at least not very much. And I'll come back to this. We do see a few of these dots down here at low levels, which occur. So these breakpoints are occurring within the N open reading frame, but downstream of the N start site, so they're not N transcripts because they would have to occur before the first AUG of the N open reading frame, and so they're potentially ORF10 breakpoints. And then on the right-hand side, essentially all we've done on the right-hand side is zoom out on the y-axis, so that instead of just looking at the first 100 base pairs, we're now looking at the first bases, I should say, sorry. We're now looking at the first 10,000 bases. And so what you see from that is, so you can see that basically this first left-hand side plot's now being compressed into just essentially a single line because we've been now every 100 base pairs. So you can see that's a single line here now, and that's where most of the activity is. But there is quite a bit of kind of read through. This is a log scale again, so but there is a bit of read through. So there's activity, what you might call an extended 5' leader sequence, and most of that activity, interestingly, seems to be immediately upstream of the ORF10 breakpoint. So that's somewhat intriguing. So that's the breakpoint analysis. And then if I could go back to the first part of this, which is the read clustering. So based on the read clustering, where we've done it by the upstream and downstream ORF, so upstream of the 5' break and downstream of the 3' break, we get these combinations essentially. So this one at the top here, the most expressed one, so I'm showing the expression levels or the depth, the total number of transcripts in the virion and the cell on a log-log scale. And so you see the most expressed one, which is over 10,000 reads in the cell data, and a bit less than 100 in the virion data, is the combination of the leader sequence, so the first 100 base pairs, plus this N open reading frame, and we saw that was the most expressed. And so this is kind of canonical, what you might call canonical subgenomes. One thing which is interesting is the spike protein, so that's the leader plus the S, which is purple. We see that we get reasonable depth, not huge amounts in the cell, but we still, we see some, but we see none at all in the virion data. So, and it's below the X equals Y line, so it's expressed at less than what we expect, although I guess the count's very low, so we can't make any really firm conclusions here. And then the other interesting thing is that as well as this canonical leader plus 3' open reading frames, we see a couple of ones arising from this kind of read through of the leader sequence. So essentially, when you look at the protein coding potential of these transcripts, it consists of the first, maybe a few thousand base pairs of the ORF1RB, plus some of the N. So not maybe the last 100 bases of the N open reading frame. So they're kind of, if these are translated, but they have coding potential, then it would consist of a kind of like a fusion between ORF1AB and N, and then the other one. So we've also got ORF1AB and ORF10, and so yeah, that reflects this kind of extended leader sequence that I showed you before. And so another way to look at this is to go back to these depth coverage plots that I showed you before, but now with these new clusterings that we have. So these are the same clusterings that I showed you on the previous slide. And so you see the most abundant in the cell is the leader plus the N open reading frame. And the vertical lines here indicate the annotated start position. The solid line is the start position of different genes, so different colors of different open reading frame, and then the end position. So you can see for this N, it lines up beautifully, the coverage with where you would expect it to start. And also these dotted black lines indicate the positions of these transcript regulating sequence. And so I guess this is the spike protein, much lower levels of expression. Yeah, so you can see how the coverage relates to the coordinates. And then these, what I was calling these kind of fusion transcripts, so there's a bit of ORF1A, B, and then a bit of the N, you can see on the left-hand side with this read through. And you can see that that's not a solid drop-off, it's basically slow decay there. And this is what we see in the virion data. We have obviously far less reads, but the pattern is broadly similar. With the expression that spike protein is not seen at all, but it's expressed at low levels anyway. And then we can do, so this is just showing you the full length of the mRNA molecule going from 0 to 30,000 bases. And we can actually then zoom in on the interesting bits, if you like. So on the left, this is showing the first 1,000 bases, and on the right, this is showing the last 10,000 bases. And I should say this is low S smooth, as was the previous plots. And so some of this strange, the smooth, strange behavior that kind of you might see here is just due to that low S smoothing. Otherwise, there's a little bit of noise and it's hard to see things, particularly on a log scale. And so you can see this read through of where we would expect. So this dashed line is where we should expect the 5' breaks to occur, but you see in some cases it kind of reads through. And then you can see that joining up to the corresponding color on the right-hand side. And then you can also see, which is interesting, in many cases, for instance, this orange one here, the break is quite distinct. So there's a single break point, essentially, or plus or minus a few base pairs. Whereas in others, there seems to be a bit of a ramp up before it gets to the jump. And the most pronounced of those is in fact with ORF10. So although if you remember originally with the histogram plot, it looked like ORF10 wasn't being expressed, but that's partly because there's a ramp up of the breakpoint. So there's no single peak where in this histogram where the ORF10 reads would lie, and instead there's kind of a distribution. And so that makes it hard to detect ORF10 from the histogram-based clustering, but easy to see here. So that's what I'm showing. Actually, the leader only plus ORF10 is indicated here in purple, and then this one here is the ORF1AB plus ORF10. So, and I guess the reason this is so diffuse is because there's no transcript regulating sequence in this area, and so it's maybe just noisy recombination that's happening here. And this is what it looks like in Virion. It's noisier because we have less reads, but you can still see this to a certain extent. It's very, in the noise though, essentially this ramp up of the ORF10. Okay, and then yet another way to visualize this data. So this is the same data as what I showed you with these coverage plots, but shown in a heat map. This is showing we've got the first 100 bases on the left here, and then we've got the first 1,000 bases, and so you can see the first 100 bases is squished up in this little area here, and the depth is showing the log of the coverage. And you can see again, this leader plus n is the most expressed. Oh, and then I should say on the right-hand side, we've got the last 10,000 bases, and that you can kind of see in many of these there's a pretty sharp cut, and others it gradually ramps up, like here, for example. So this is just essentially another way of visualizing all of these, of both the canonical and non-canonical subgenome mRNAs. And as I said, the spike protein, we don't see any of that. So that was from the cellular derived RNA, and this is from the virion derived RNA. We don't see any of the spike, but it's fairly low expressed in the cell, a bit lower than the others, but it's lower than what we'd expect in the virion data. It would be good to have more data so we could confirm that it was actually absent. So the other thing with nanopore direct RNA sequencing is that you might then start to look at the signal level and see if you can identify modifications. And so there are programs obviously for doing this, such as Tombo, which for 5MC at the moment, and EpiNano looking at M6A. So we've done a little bit of Tombo analysis, which I won't present here, but I just wanted to instead show you the base calling error rate. So papers have shown that for certain modifications, one of the signatures you see in the data, and I should say this is not necessarily at the read by read level, but overall when you have high coverage of reads, is you see a greater base calling error rate. And so this is showing you the same coverage plots I showed you before. So for the cellular RNA, so less than 100, and then less than 1,000, and then greater than 20,000 on the right-hand side. But instead of plotting the coverage, I'm now plotting the proportion of mismatches and deletions, essentially on a scale of 0 to 1. So when you see white, means there's no coverage. When you see yellow, it means it's close to 0, so that there's a very low error rate, if you like. And then when you see a red or an orange or a red, it means there's a higher error rate. And so, I mean, there's some intriguing patterns here, I guess. It seems like, when you do read through Well, yeah, I mean, when you go past 100, so this is 100 base pairs down here. So when you go read past 100 bases, it seems perhaps there's a bit more base calling error. Whether that reflects for RNA modification or not, we're not exactly sure yet. You can see that on this right-hand side where there is a slightly elevated rate of error, that seems pretty consistent across all of the subgenomes. And when we looked at the M5C modifications in Tombo, we also saw a very high degree of consistency, and whether that just reflects the fact that errors are more likely to occur at certain k-mers than others, whether it's a genuine modification, that position is still a bit of an open question at this stage. But you can see there's some intriguing patterns. And so this pattern here on the left is quite intriguing. It seems to be consistent between some of the subgenomes, but absent in others. So, you know, there's potential for subgenome-specific modification. That's in the cellular RNA. And if we have a look at the virion RNA, there's less coverage, so we can be less certain. But I guess there's some intriguing, you know, similarities and differences here as well, I should say. But this certainly needs to be explored in greater depth, and we're hoping to do that. The other thing we noticed is that overall, the virion RNA actually has a higher error rate than the cellular RNA. So this is just showing for all of the subgenomic RNA we identified, so only those which are canonical with the canonical leader sequence, just showing the distribution of the error rates across all reads. So this is what I'm plotting is I calculate that error rate for each read in each cluster, and then look at the distribution of those reads indicated by a violin plot here. And so you see the cell in cellular material in orange has substantially less error than the virion-derived material. And so we speculate that may be due to modification, a high level of modification on the virion material, but obviously we need to do more work to confirm that. We also can look at the poly-A length. And here, this is in the opposite order to what I presented here. So this is going from the shortest to the longest. So bin 1 actually corresponds to the N open reading frame. And we're just plotting the distribution of poly-A lengths across all reads in that cluster. And then the right-hand side of the reads that don't have a leader. And these kind of by eye look fairly similar with the exception of the N open reading frame. There seems to be a big cluster here of N open reading frame transcripts that have very short poly-A length. And another preprint has suggested that there's a correspondence between the length of the poly-A in this cluster and the degree of modification of those molecules. So I'll just conclude here and say that direct RNA sequencing provides insights into transcriptional activity of SARS-CoV-2. So unlike the tiled amplicon strategy, which is much better for finding mutations, this kind of shows you what's going on transcriptionally, including identification of subgenomic mRNA, poly-A links, and potential modification sites. So ORF10 is an interesting one because it has no kind of protein homology. However, you know, at first we thought it wasn't expressed based on the histograms, but a finer inspection indicates that it is expressed. We can find around about 60 transcripts in our data. So that to me gives me confidence that it's real, but it's at low levels. Seems to be a much smaller proportion of subgenome linked mRNA in the virion data, and also the high error rate of the virion data is something that suggests perhaps there's more RNA modification going on. Just in comparison to SARS-CoV-229E, SARS-CoV-2 does seem to be slightly more complex in its transcriptome in terms of the number of open reading frames. And all of the analysis we've done, we've developed a package which everyone can, if they want to, download. It's a combination of Java for calculating doing all the clustering and calculating the breakpoints, and then R for visualization. That's in this GitHub repository. And so I'd just like to acknowledge all the people who are essential to getting this work done. So particularly the Victorian Infectious Disease Reference Laboratory. So they grew up this virus extremely quickly and distributed around the world, and also made some of the sample available for RNA sequencing. So Julian, Mike, Leon in particular. And then George was really instrumental in driving this project forward and kind of had the initial idea to do this, and working with Leon to get the material, and George did the sequencing on nanopore as well. So George really deserves a lot of credit for driving this project forward. Daniel Rawlinson in my group did a lot of the analysis. George is part of Deborah Williamson's group, who provided a lot of oversight. Damien Purcell, the virologist, provided expertise in understanding what the data meant. And then also on the paper, Sebastian Duchenne and Leo Featherstone did a lot of the evolutionary analysis, which I haven't discussed at all. And Miranda Pitt assisted with the RNA sequencing. So we'll finish there and take any questions. And I can see there are a few questions, so I will just go to those. So the first question from, let me just make sure I'm getting all of these. Okay, so from the data, is it possible to measure the viral load? So there are other ways of doing this, which don't require sample sequencing. So yes, it would be possible, because you also get the human RNA, obviously, as well, which we haven't talked about at all, but we're also looking at that. So when you do the direct RNA sequencing of the cellular material, you get quite about maybe half to 2/3 of the data is actually mapped to the human genome. And so I guess you would be able to get some idea of the relative amount of viral RNA and human RNA, and then you'd have to scale that by the size of the transcriptome, I guess. So you could calibrate it if you wanted to. Thanks for that, Nick. Seeing if there's any other questions. Oh, I've got 12 questions. Okay, I'm trying. Okay, I've got a question from Teruhito Ishihara. Are there any RNA virus-specific RNA modifications? Right, so yes, we think there are a number of modifications. So in our preprint, we suggest there were 42 sites of 5NC modification. I'm not sure what you mean by virus, you mean specific to this coronavirus and not others. I guess we haven't tried to check. I mean, we've only got this other direct RNA data set for comparison from this HCOV229E. So, and also, homology is not that great. So it might be hard to say to what extent the same base is either modified or not, but we should certainly try and investigate that. A question from Chloe about de novo assembly. Actually, that was our first instinct, was to do de novo assembly, but actually, so colleagues did a de novo assembly of Illumina data, and actually that was a perfect assembly. Like, there's no gaps. So it's kind of viruses are easy to assemble. You don't really need nanopore sequence data to assemble it. And also, as you might have seen, our coverage is very highly biased to the 3' end, so the coverage really drops off as you get closer to the 5' end. So it would be excellent for assembling the 3' end, but wouldn't be so good for the 5' end. But the assembly was so good anyway. I mean, it's not like bacterial genomes where you need long reads to complete the assembly. It was a perfect assembly from Illumina data. I should say that, yes, go on.
Can I just interrupt for a second, Lachlan? Just might help to remind everyone how to ask questions. But we mentioned also about putting a hand up, which I actually can't see now how to do that. So you might just tell people if that's obvious. But also, I'm not seeing the question, so I guess no one else is either. So just make sure you read them out fully. Yeah. Thanks.
Yes. Sorry, I can see there was one hand raised, but now it's gone down. Yes, okay. So the next question from Belinda Lin is, do you think transcriptomics affects the sensitivity of the PCR assays on the market as they detect a combination of E, N, or RD/RP gene? I see. I guess it would be better to try to detect a high expressed gene because you've got more molecules to work with. The N is high express, so it's good that they're going for that one. I mean, I think if I was designing an assay, I would go up to the N, given what I know about the level of expression coverage that we got. So that seems to be the right thing to do. Then question from Olin Cylinder: how much different in transcript structure do you see in cDNA versus dRNA data? Right, so we haven't got direct cDNA, we haven't got long read cDNA data, and that's something we're planning on doing. We've gone straight for direct RNA, but it would be beneficial, we think, to have cDNA data as well, because then you still don't, I mean, you have to do the second strand synthesis, but you still don't have the amplification, so it'd be good to compare and contrast those two. Then Yuan Cheng asks, do you have a solution for direct RNA cDNA sequencing that's independent of poly A? Right, so that's another thing we want to do. Miranda Pitt has done direct RNA sequencing on bacterial transcriptome, which don't have poly A, and that she did that by essentially adding the poly A to everything. And so that works quite well. And so actually we're thinking about doing that, even though of course you do have the poly A, there might be some transcripts from the middle which don't have poly A. So it might be interesting to look at that too. Okay, so I think that's all the questions, but if anyone feels like I didn't answer the question properly, please ask a follow-up question. No one's got their hand raised. If anyone wanted to make a comment or say something, they could put their hand up. So someone has got their hand up now.
Just remind me where you do put your hand up, because maybe my screen's different, but I can't see that.
To be honest, I'm not sure that.
Maybe I'm seeing something different. Yeah.
As the speaker, I'm not sure.
Yeah, maybe we can't do that.
Yeah.
Okay. Well, I hope it's obvious to everyone.
Someone had their hand up, but now it's gone down. That's fine.
But maybe I can just ask a naive big picture question while, because obviously, you know, this is very far from my work. Well, obviously, we're all kind of thinking about the sort of urgent efforts to control the spread of this virus. I mean, can you foresee any way that this, you know, detailed level understanding is going to feed into, you know, control strategies?
Yes, that's a good question. Also, the other preprint I mentioned, where they did the proteome map, which is, you know, a bit closer to function, if you like. Like, you really want to be looking at proteins if you're going to design antibodies or drugs. And so knowing which proteins are expressed would be useful for doing that, I guess. So I think coupling this with proteomics, which is in fact one of the papers I showed earlier, does, and then the other paper which did the protein interaction map between human proteins and SARS-CoV-2 proteins would be also very useful data for designing drugs. I can foresee use of direct RNA sequencing for monitoring response, if you like, and for also, if you do find something that, so for instance, if it does turn out that chloroquinolone acts against the virus, then actually seeing what that does might be useful. So I can foresee kind of using this in combination with different treatments might be of use too. But I guess in terms of the findings I presented today, I mean, I see it as kind of understanding the biology and perhaps not yet directly indicating how we might control it, but hopefully someone else can make the link to do that. Okay, so I've got some hands raised. I might go to one of those, and I've still got more questions. I'll come back to them too. So let's see if I can do the hands. Oh, here we go. So Anubhav, I'm gonna, hang on, did that work? So the hands have gone down. Sorry, I think I just figured out how to unmute people. Okay, Roy. You're on, Roy.
Oh, hello. Hi, Sir Lachlan. I was wondering if about your opinion, if the universities in Australia should get involved into testing, and what will be the pros and cons on that?
Right, so I mean, it does seem to be the case. This is kind of with our area of expertise, but it does seem to be the case that the more testing, the better you're able to control the spread of the virus. And so definitely increasing the rate of testing seems like a good idea. And maybe that doesn't need to be done in university labs, but I guess if there's lack of capacity, then it might be a good idea. Certainly probably having people who are trained to do PCR assays come and help out with that seems like a good idea. And I know at the Doherty, for instance, they put out a call for people working at the Doherty who are kind of on lockdown if they want to come in and help out in the diagnostic lab to try and improve the rate of diagnostics. Okay, so some more questions. Okay, so and above, there are reports that the viral genome is relatively stable. Have you done comparative analysis using sequences? Right, yeah, this is an interesting question. So as I mentioned, there are 3 other direct RNA sequence data sets available. And people have also compared the genome sequence you get out from the tiled amplicons as well. And yeah, it does seem relatively stable. The mutation rate seems like relatively low. But I guess the other point is we're looking at cellular derived material rather than patient derived material. And we did see in our sample that there was a 10 base pair deletion in the UTR, which seems to be specific to the cellular data that we had. And we also found was in a number of other cell line data sets in other Australian samples. So there may be some, we speculate in our preprint that there may be, can tolerate deletions in a permissive cell line, which might not be able to tolerate in surviving in a human host. Yeah, so yeah, perhaps it would be less stable. I mean, perhaps it's stable because it has to be, but it's got an error correction. As I understand, it has error correction enzyme as well, so that's one reason why it's quite stable. Have I checked? So Chloe Goldsmith asks, have you checked with other callers for methylation like Guppy and Medaka? Yeah, right. So yes, we have done. We are working on that. That's definitely a priority now. We've done, as I mentioned, the Tombow was the main one we did, and that's mostly because we wanted to be able to compare to the analysis presented in the genome research paper I presented at the start with the Digitombo analysis. But yeah, we're working on using other techniques, and I know Eva Novoa in Barcelona is going to apply EpiNano to this data set and others, and so we're hoping that will shed light on the M6A modifications. Belinda Lynn asks, where can we read about our work? So I mentioned everything I presented is on bioarchive. Well, not all the data, not all the slides I presented are on bioarchive, but I presented the story around a paper which is on bioarchive with a few extra analyses, which will go into an updated preprint shortly. So if you search for my name on bioarchive, I guess you should find that. So then Chi asks, can you remind me what is the difference between the leader sequence and sequences and others you have in the sample? I'm not sure I quite understand that question. So we just have, we have a single, in the data set I presented, there's just a single virus, both in the virulent state and the cellular state, and the leader sequence is essentially the first 80 base pairs or so. And Jessica asks, I was wondering if you could post references using your talk. Yeah, I'm happy to put the talk on Figshare, so I'll put it on Figshare afterwards. Roy, oh, you asked a question about testing, thanks. Oh, George, thanks for tuning in, George. So yeah, George Tarara, who's the first author on this study, is kindly asking me a question. And I didn't plant this, but I could have. So are you interested in exploring the host transcriptional response to SARS-CoV-2 infection? Yes, absolutely. So as I mentioned, about half to 2/3 of the RNA that you get from the cellular material is actually from the host. And so we've been looking at that. And as you know, it's just an N=1 sample at the moment, but it'd be interesting to look at larger sample sizes and compare infected to uninfected. So that's definitely something that we would like to do. And also, yeah, I guess look at how things change over time as well. Mark Schultz asks, do you see any HEC calls indicating mixed or within host infections? Right. Actually, George is probably the right person to answer that question. I'm not sure if you have a comment to that, George. I'm not sure about that. Yeah, okay. So I will unmute George and he will be able to answer that question. Okay, George?
Hey, is this working?
Yep.
Okay, so yeah, we do. Even on the direct RNA, there's some hits. So it looks like there's some quasi-species, and not for every sample, but definitely for a few. And that also appears in some of the Illumina data for these cultured isolates and some from the amplicon work as well. So yeah, they're there. We're not quite sure what to make of them yet.
Cool, cool. Are they, do they change the protein, the amino acids in the protein at all?
I haven't had time to dig into them personally, but I think other people are following up with it.
Yeah, cool, cool. I'd imagine they're easier to detect in the N-ORF because it's much higher levels of expression than, say, the spike protein, which is a relatively low level of expression. So Rick DePauli asks, what was the original animal before transfer to humans? Yeah, that's a big debate, obviously. So, I mean, people have shown that genomically this has most similarity to bat coronaviruses, but it's not known whether there's an intermediate host. And yeah, I think a lot of people, I saw a paper on bioRxiv which suggested that cats can be infected with SARS-CoV-2 and that they can infect other cats as well. So, but pigs and dogs can't be infected, or at least it won't replicate very well. So I'm not sure what that tells you, but be careful if you've got a cat, I guess. And then Stefania Bertazzoni asks, how long is ORF10? And also you said it's got no sequence homology. Is this true for an HMM profile? So, right, so if you put it into BLAST, how long is it? That's actually a good question. Let me just check need to check, put the file here. So it's 117 bases, it's pretty short, 117 bases, so 40 amino acids, I guess, almost. So that's very short. And yes, if you put it in BLAST P, you don't find a homology. So if you search an HMM profile, I've looked in PFAM, which is, you know, the group of HMM profiles, obviously, and didn't find anything there either. But of course, if you have a better HMM profile, you might be able to find something, but not in the kind of known gene families, for example. Yeah. I've noticed that PFAMs, yeah.
Just finish what you're saying. Yeah.
No, PFAMs have recently released a new SARS-CoV-2 set of HMMs, so it'll be interesting to try that. But I guess now we've seen it, and we might just find matches to that alone. We want to find matches to something else, but we can't yet. Okay, David, sorry.
Okay, so I was just going to, you know, we passed 1 p.m. and obviously, you know, quite a number of people have had to leave already, and so maybe I'll just interrupt now. Take this opportunity to thank you. Thanks for being the guinea pig of our first online seminar. I hope there'll be more to come, but I think it's worked well and attracted a lot of interest. So that's great. Thank you for paving the way.